compromised, and those particles that were already reached the tumor and cannot be removed. And this is also called permission, so in retention. So altogether is enhanced permeation and retention, which is really the basis for the targeting to the tumor and by nanoparticles from now. However, recent study seem to suggest that in the human tumor, in a solid tumor in a human patient, and the EPR is much less dramatic, it's much heterogeneous and the phenotype. And that a lot of the nanomedicine we have developed might be too large, to benefit from the EPR in a human patient. So this is a real question, how what we are doing now can be translated into the clinic, and also we know that this EPR and largely applied to the primary tumor, and for the disseminated or the small metastatic lesion, which do not have a good the blood supply, and the EPR and may not work. So the last 5-10 years effort in my lab has been focused on developing two strategies and to overcome this major challenge. The first strategy is to develop a very small size particles we call ultra small size nanoparticles. So the idea is that even though the chemistry in a human tumor is small, but if our particles are smaller, then they can still reach the tumor and through the EPR. And the second strategy is to target to the tumor endothelial cells and actively and can reach the tumor and through this transcytosis. And I will first and talk about the first and the strategy, and which is the ultra small nano carriers for co-delivery of hydrophobic and hydrophilic drugs. So we started this work when we trying to achieve and co-delivery of the paclitaxel and the gemcitabine for the treatment of pancreatic cancer. As many of you know that this is the frontline treatment and for the pancreatic cancer, but the efficacy is limited, by the limited bioavailability, and also the dose limiting and toxicities. And so we need to develop a nanocarrier to improve the deliveries. The paclitaxel is poorly water soluble, and the gemcitabine is highly water soluble. So the co-formulating of these two drugs of different physical property and using liposome vector has been very, very challenging. Another strategy is to covalently conjugate both drug into a polymer, but this will involve complicated conjugation chemistry, and also it's very difficult to control the ratio of these two drugs and without compromising the delivery function of the polymer. So Gina Sum, they joined my lab a couple of years ago, and she's currently assistant professor at the University of Nebraska. So she decided to conjugate the gemcitabine and to the polymer, and then use this gemcitabine and conjugate the polymer and to co-load and deliver the paclitaxel. So the polymer and she used is called a PVD. Again, this is amphiphilic and the polymer can self-assemble and to form the particles, but the size are relatively large, about 150 nanometers, and also the drug loading capacity is limited. So the gemcitabine is a nucleoside and analog and has a sugar group and with several hydroxy group. So I will come back and talk about this again. So the gemcitabine and conjugate polymer is essentially, we call it PGM, is essentially a gemcitabine prodrug, and it has a biological activity when this is cleaved at the tumor tissue. But at the same time, we can also use this to co-load and deliver another drug, and such as the paclitaxel. And interestingly is that the conjugation of gemcitabine to the PBD led to a drastic decrease in the size from about 150 and to about 10 nanometers. So we know there are many strategies, for the size reduction, but the problem is that when you make the size too small, they can no longer load the drug anymore. So surprisingly that instead of seeing another decrease in drugging loading capacity, we saw significant improvement in the performance of the PGM in loading a number of the drugs that we have tested, and this is only a short list of the drug, and we have tried, and for the paclitaxel, and it can reach about 20-fold percent, for the drug and the loading capacity. So we are quite excited, about this potential application, for the treatment of pancreatic cancer, because this cancer is notoriously known to have a dense and stroma, and the accumulation and the penetration and has always been an issue. And we saw that PGM might have an advantage because of its very small size. And so this is looking at the distribution of fluorescence dye that is loaded into the PGM or the PBD and the control particles. So this mice, has two tumor and one on each side, and this is an ex-vivo image. And you can appreciate the significant improvement in accumulation of the small sized particles at the tumor site. So when we look at the distribution, under the microscope, and you can see the wide the spread and the distribution, and with the PGM and the particles. And for the PBD particles, the particles are largely located in the peripheral an area of the tumor which is adjacent to the blood vessel, suggesting that these small-sized particles are not only effective in accumulation, but also in the deep and penetration. So this is the subacute tumor models, and we did similar studies with also topic models, and we also saw significant accumulation of nanoparticles in the tumor tissue. So this is looking at the therapeutic efficacy of the PGM alone and all the PGM and loaded with paclitaxel. And this is the PDX model, and this is a Myount model of the colon and the pancreatic cancer. And interestingly, the PGM itself is quite ineffective. Actually, it's even more effective than the combination of the taxol and the free and the gemcitabine. But the co-delivery of the paclitaxel and the gemcitabine and using the PGM nanoparticles led to a further improvement in anti-tumor efficacy in both the PDX model and also the Myount and cancer model. So as I just mentioned, the PGM itself is quite effective, and we want to study the mechanism. And so we are looking at changes in gene expression profile after treatment of the PGM, and this is the RNA-seq data. And as you can appreciate, treatment of the PGM led to the inhibition of a number of the oncogenic and the pathway, which is in agreement with its potent anti-tumor activity. And interestingly, the PGM can also activate the type 1 interferon and the response, and also the STING and the pathway. That we know that there is a lot of interesting in boosting this the pathway for cancer immunotherapy, but it's limited by the systemic and the toxicity. But at the same time, we also notice some undesired and the response and such as the induction of CCL7 and the CCL2. So this is the RNA-seq data. So and we also confirmed the induction of CCL2 and CCL7, and both the in vitro and also the in vivo. So what are these two molecules? And CCL2 and CCL7 are the important and the chemokines from the. So they can be produced by both tumor cells and also the stroma cells, and they can come back and act on the tumor cells and promote the tumor cell proliferation and the metastasis. But more importantly, these and the chemokines can recruit a lot of the immunosuppressive and the immune cells, such as myeloid-derived suppressor cells and the Treg cells, and also the M2 and the macrophages. So this raises two questions. First, what is the mechanism of this chemical induction? And the second, whether we can combine the PGM and with the CCR2 and antagonist, and because both chemokines and they share the same receptor, which is the CCR2. So we have done a lot of mechanistic studies, which is published, and then already I will not get into all the details. As I just mentioned, the PGM can activate the STING, and the STING can stimulate the type 1 interferon response, which is good for anti-tumor immunity. But interestingly, STING can also cause some undesired response, such as the induction of CCL2 and the CCL7. So if we combine the PGM with CCR2 antagonist, we might be able to maintain this favorable response, but it can inhibit this induction of CCL2 and the CCL7. So the PF is CCR2 antagonist developed by Pfizer, and as this is a non-metastasis model, and as you can see, the PGM itself is still quite effective in inhibiting the metastasis, but the combination of the PF and with the PGM led to a further improvement in anti-tumor activities. So if we're looking at the immune profile in the tumor tissues, we saw significant decrease in the infiltration of the immunosuppressive cells, such as the MDSC or the M2 macrophages. At the same time, can promote the recruitment of the good immune cells, such as the NK cells and also cytotoxic the T cells. So how can we form such a small size and particles? And we have done a lot of mechanistic study and based on the computer modeling and also the structure and activity and the relationship study. And this is published in this paper. And so this is our model. So like many other amphiphilic molecules, and the PBD can also self-assemble to form micelles. Initially, this particle size is very small, but they are not dynamically stable, and they tend to undergo the intermolecular micellization and form a much more stable but relatively larger size in the particles. So remember, I mentioned at the beginning that gemcitabine is a nucleoside analog that has a sugar motif and with an hydroxy and a motif. So for this PGM, after the formation of the small size particles at the beginning, these and the hydroxy group, we have a lot of hydroxy group that can interact with the surrounding water and through hydrogen bonding, and this can help to stabilize these small sized particles. And at the same time, some of the hydroxy group may interact with the loaded drugs such as paclitaxel and through the hydrogen bonding. And this can not only improve the drug loading capacity, but also help to improve the stability of the drug and loaded micelles. So we know that there are many nucleoside analog that share, the similar structure. So we can actually conform the ultra small size nanoparticles by modification and with a different nucleoside analog. So we have developed a small size platform and for different combination therapy. And for example, the cytarabine is a backbone drug and for the acute and myeloid and leukemia, and it's often and combined with another drug and such as the daunorubicin. So we also did a similar studies and in the acute myeloid leukemias and in collaboration with Dr. We do in our human cancer centers. So we did lot of studies on the transgenic models and also the PDX model. And due to the time, I will not get into all the details. So to summarize, the first part of my talk, we have shown that the polymer modification with the nucleoside drugs led to the formation of ultra small size particles that are not only effective in drug loading, but also very efficient in tumor accumulation and penetration. And we believe, and this is a platform and it can be used for different and combination therapy. And we also believe that in addition to the improvement in the formulation, we also need to understand, the mechanism of action of our nanomedicine, and this can help to improve the overall and therapeutic and outcome. Now I'm going to switch gear, to talk about the second strategy, and which is to actively target the tumor endothelial cell and reach the tumor site and through the endocytosis. So our initial and system and was developed to address a need for cold delivery of SKRA and the chemotherapeutic drugs, and to overcome the immune resistance that happens after immunotherapy. So what is SKRA? I think we first need to talk about phosphorylserine, and which is also called under the PS. So we know that our bilayer cell membrane is composed of a lot of phospholipids such as the PC, PA, and the PE, and the PS is another phospholipid. Interestingly, the distribution of the PS is not symmetrical across cell membranes, and most of the PS is restricted to the inner leaflet of the cell membrane. And this asymmetrical distribution is regulated by group of the flippase and also I feel the scramblase. So when the cells are under the resting condition and the scramblase are not active, and this flippase will constantly bring the PS from outside and to the inside. So that's why we saw a lot of the PS as shown by these the red dots. But when the cells become activated, such as activation of the platelets and the increased intracellular and calcium, and will cause inactivation of the flippase, and at the same time will activate the scramblase. When the scramblase is not active, it will randomize the distribution of the PS across the membrane and cause more exposure of the PS on the cell surface. But this is a reversible process, and when the calcium is removed, and the PS will restore to the normal state of the distribution. Whereas another scenario where when the cells become apoptotic, so we know when cells are dying from apoptosis, and the caspase will become activated, and the caspase actually can cause cleavage of the flippase and can cause a permanent activation of the flippase at the same time, and will cause inactivation of flippase at the same time, and will cause the permanent activation of SKRA, and will stimulate and cause the exposure of the PS on the cell surface. So different from this process, and the PS exposure in apoptotic cells is permanent. Previously, it was generally believed that the SKRA is constitutively expressed and just waiting for the activation by the caspase. As I will show in the coming slides, we have shown, at least with the cell line we have worked with, that the basal level of SKRA is very low, but it can be significantly induced by chemotherapeutic drug. So when the cells exposed to the surface of the cells, this has a profound biological consequences. So what happens is the PS can serve as a eat-me signal and can invite and macrophages and for the phagocytosis. The engagement of the PS on several and PS receptor can have a profound immunosuppression and through the stimulation of the highly immunosuppressive and cytokine such as TGF-beta and IL-10. So tumors are very, very smart and they can hijack, this mechanism. So we know that when we treat the tumor with chemotherapy, a lot of tumor cells will die from apoptosis. And those dying apoptotic cells will expose PS on the surface and cause immunosuppression. This can help to protect the remaining and the live cells, and this is a major mechanism and for the cancer relapse, after the chemotherapy. So the discovery of the PS and the signaling has led to a number of the strategy and to overcome the immunosuppression. And one of the strategy is to block the signal pathway. As I mentioned, the PS is engaged with a number of the receptor. So blockade of one of the receptor and may not be effective. And the second strategy is to use PS and the neutralizing antibodies such as PS antibody. And this approach actually has a problem because this antibody does not recognize the PS. It actually recognizes PS and the recognizing protein in the serum, so that the effectiveness of this approach is dependent on the level of that serum protein. So our strategy is to directly target an SKI8. We did not work with SKI8 before, and we become interested in SKI8 when we try to study the mechanism of the chemoresistance in the murine and cancer models and after treatment with 4-OXP. 4-XP is a pro-drug conjugate of 5-U and oxaliplatin. It is a highly lipophilic, can be readily loaded into the micelles and particles. So in these studies, we show that after the treatment by the 4-XP and the TCF was significantly induced. And this is a well-known oncogene and that is involved in the tumor progression and also the drug resistance, and kind of suggests that our approach, screening approach, might be working. And interestingly, an SKI8 is another gene that is significantly induced after the treatment, and this was discovered, and by the Yuan Chen, who just recently graduated from our lab and now joined Dr. Anderson's and Bob Langer's group at MIT. So this is the RNA-seq data, and so we also confirmed the SKI8 induction and by the RT-PCR and also at the Western. As you can see, the basal level of SKI8 is very low, and is significantly induced, after chemotherapy treatment. So what does this mean? So if we can block the induction of SKI8, we might be able to block the chemotherapy drug-induced and PS exposure, and this can help to overcome the chemotherapy-induced and immunosuppression. So there's no small molecule inhibitors of an SKI8, and we develop a co-delivery system and for the delivery of SKI8 siRNA and also the 4-XP. So this is the new systems that is developed by a senior chemist and the Yixiong Huang along with the Yuan Chen. So this is an amphiphilic the polymers and can self-assemble into the particles into which we can load these lipophilic drugs. And we also have a positive charge on the surface and because of this guanidine group, so they can form a complex with siRNA and through charge-charge interaction. So at this stage, we still have excess and positive charge on the cell surface, which is not suitable for systemic administration because we have a lot of negative charge protein in the serum. So we then converted the positive charge surface to a neutral or a slightly negative charge surface, by coating and with a negative charged and polymer, which is and controlled and sulfate. So this is and a highly and negative charged molecules, but it is also a natural ligand and for the CD44, just like a hyaluronic acid called HA. And we know that the CD44 is overexpressed and in tumor cells and also the tumor endothelial cells. So through the surface degradation and with the PS, not only we can convert the surface and the charge, we also introduce an active targeting and functionality. Well, the CS-mediated targeting and by the HA or the CS and it has been studied for decades. And one of the problem is that the CD44 and is also expressing a lot of other cells, particularly the liver sinoendothelial cells. We know that the liver is huge and has a lot of sinoendothelial cells. So without the surface modification, most of the nanoparticles will be removed by the liver sinusoid and endothelial cells because of its abundance. If we decorate, the surface, this in this cartoon, this the blue color is the CS, and the yellow color is the PEG derivatized CS. So if we modify the surface and with PEG, it can inhibit the binding of the nanoparticles to both the tumor endothelial cells and also the sinusoid endothelial cell. But because the tumor cells and the tumor endothelial cells, they express a much higher level of the CD44, if we use an optimal amount of the PEG and the modification, we can significantly inhibit the binding to the sinusoid endothelial cell and without compromising so much the binding to the tumor cells or the tumor endothelial cells. So we believe through this and the modification, we can significantly inhibit the binding to the sinusoid endothelial cells and will release most of the nanoparticles to the blood circulation, and they can effectively interact with the tumor endothelial cells, and eventually will reach the tumor and through the transcytosis. So these slides, summarize many years of the formulation and optimization work, and there was a lot of disappointment, at the very beginning, and we are very happy that we finally, identify an optimal and formulation. So this is looking at the distribution of fluorescence and labeled an siRNA, and we know that the free siRNA and cannot reach the tumor because of its rapid degradation in the blood. If we form a positive charge complex with a canonical polymer, they will be largely trapped in the lung and because of aggregation. If we decorate the surface with a negative charged molecules called polyglutamic acid, and we can decrease the lung accumulations and increase the targeting to a tumor, but we saw a lot of the uptake in the liver because of the interaction with by the liver cells and also the sinoendothelial cells. But if we coated with the CS to include and to introduce the targeting abilities and whereas significant increase in the tumor targeting, but also has a substantial uptake in the liver, and we believe this is due to the significant uptake by the liver sinusoid and the endothelial cells. But if we introduce an optimal amount of the PEG, whereas a drastic decrease and in the liver uptake, and along with a significant improvement in the tumor targeting. So we did this study in many tumor models like breast and colon cancer models and the PDX model and also the orthotopic models and this system also work for the lung metastasis. And as I mentioned, the EPR and does not work for the micro and the metastasis because they do not have a good good vasculature and the supplies from there. So in these studies, we show that our system can effectively target to the lung with metastasis. If there was no metastasis and there was no accumulation. So we believe that this is achieved through active targeting and the mechanism that is independent of the EPR. So this is and shows you the tissue distribution and of the proteins and all the siRNA in the tumor and also several other and major organs. And as you can see, the accumulation in the tumor is much higher in all of the other organs, and we can achieve about 10% of the injected dose in the tumor and tissue. Well, as I mentioned, the CD44 and it has been studied for several decades, and we know the CD44 is important. But the CD44 is expressed in both tumor cells and the tumor endothelial cells. So what is the role of the CD44 on the tumor cells and also the tumor endothelial cells in the targeting and the penetration of nanoparticles, and it has never been well studied. So this is a work that is done by a talented and student, and Dr. Zhang Yinluo, and who is about to graduate, and now he's looking for his future lab. So in these studies, and he first generate the CD4 knockout tumor and using the CRISPR and technology, and he also purchased the CD44 and the knockout mice to mimic the CD4 knockout on tumor endothelial cells, because we know that the endothelial cells is coming from the mice. And then he developed both the white type tumors and also knockout tumors in either the white type mice or the knockout and the mice. And he looked at the distribution of the nanoparticles. So interestingly, and if we knock out the CD44 and on the tumor cells, there is only a small decrease in the signals of the nanoparticles. But if we knock out the CD44 in the mice, there's a drastic decrease in the signal of the nanoparticle, both in the tumor and as well as in the liver. Then when he looked at the distribution under the microscope, and he found out that if he only knock out the CD44 in the tumor cells, and we still have a lot of signals, but the signal is largely located in the peripheral area, and that is close to the blood vessel, and as shown by the CD31, which is an endothelial and the marker. But if we knock out the CD4 in the mice, and there is a drastic decrease in the signal of the nanoparticles. So these studies seem to suggest that the CD44 in the tumor endothelial cells play a very important role in determining the amount of the particles that can reach the tumor site, and the CD44 on the tumor cells and play a more important role in the penetration. And to test whether the transcytosis play an important role in the penetration, so he did the transwell studies. So this is the tumor cells, and the nanoparticles was applied to the upper chamber, and then he collected the nanoparticles from the lower chambers. And as you can appreciate, that the CS coated nanoparticles are highly effective in passing through the tumor cells layer and through the transcytosis, and this can be significantly inhibited by the transcytosis inhibitors. And this event can also be abolished when he knocked out the CD44 and in the tumor cells, and he found a similar result. And when he did the experiment and with the tumor and the endothelial cells. This is the tumor and the sphere and the penetration studies and as you can appreciate, that the CS-coated nanoparticles are effective in penetrating the tumor and reach the core of the tumor, and this process can be inhibited by either transcytosis inhibitor or the knockout of the CD44. So taken together, this is our proposed models from so after the particles bypass the liver and reach the tumor and the circulation, and most of the nanoparticles will bind to the tumor endothelial cell and will reach the tumor through transcytosis. And a small number of nanoparticles may actually reach the tumor and through EPR. And upon reach the tumor, and this process can continue, and some of the drugs will be released, and the other drug will reach the other side, through transcytosis, and this process will continue through several layers of tumors. So we believe that the CD44 on the tumor cells play very important roles in defining the amount of the nanoparticle that can accumulate in the tumor, while the CD44 on the tumor cells play a more important role in the tumor penetration. So this is the efficacy and the studies, and we can show that the chemotherapy alone does not work so well, and the SKI nanoparticles and also only have a modest effect, and the combination of the two and the net drastic improvement in the anti-tumor efficacy in both the murine and also the pancreatic and the cancer models. And this is is associated with a significant improvement in the tumor and the microenvironment, including the decrease of the immunosuppressive cells and also the increase of the good immune cells. And we also did the combination and with anti-PD-1, so and we can achieve a significant improvement in anti-tumor activity in the advanced tumor models. So we started the treatment when the tumors are already relatively big, and we also can achieve a long-term survival and with this combination. So this is done and with the subQ models, and we also did the similar studies and with and also topic and the pancreatic cancer model. So these the tumor cells express luciferase and then can be monitored by IVS. And as you can see, this combination was much more effective in inhibiting the tumor growth in this topic model. So this is our proposed models. So if we only treat the tumor with a chemotherapeutic drug, and we can kill most of tumor cells, but these tumor cells and also have PS exposure on the cell surface, and this can cause they can interact with the macrophages, and then cause release of immunosuppressive and the cytokines, and can cause an immunosuppression and the relapse of the tumor. So if we co-deliver the chemo drug and with SKIA, and we can effectively block the induction of SKIA, and we can inhibit the PS exposure, and we can prevent this immunosuppression. So if we prevent the phagocytosis of apoptotic cells by the macrophages, and these apoptotic cells will undergo the secondary necrosis, and this is a much more pro-inflammatory the process, and can stimulate the tumor immune microenvironment, and will lead to a more favorable and outcome. So to summarize, the second part of my talk, and so we have identified the SKI8 as a new therapeutic target, and we also develop a new system, and that can be used for delivery of chemotherapeutic drug alone, and SI alone, and also the co-delivery of both drugs. And we believe that this is works through a mechanism, and that is not solely dependent on only EPI effect. And the targeting an SKI8 in combination with chemotherapy, and represent a new immunochemotherapy approach for the treatment of different type of the cancer. And so I'm very grateful for having a very hard working group, and this is some of the people in my lab, and also my research has benefited from the collaboration with a lot of investigators and inside and outside the Pittsburgh, and this is and the source of the funding for our current work. I also would like to thank you and for your attention, and I'll be happy to answer any question.
Okay, thank you very much, Dr. Lee. So we have a few questions in the chat. While you're answering, we have 10 minutes at least for questions. Please feel free to type in more questions, and Dr. Lee can answer. So first question is from Joubert Johnny. asking about what causes ultra small particles to be retained inside a tumor.
Yeah, this is actually a very good question. So this is the slides we prepared before the submission and of another manuscript. In fact, and actually, for the ultra small size nanoparticle, the mechanism is not simple. And I cannot disclose too much information, but I can share with you some information. So what happens is like these ultra small size nanoparticles, and they actually can recruit some serum protein. And so by modulating the geometry and the size of the particles, you can actually modulate the pattern of interacting with certain protein. So this and small size particle, they can recruit protein that can mediate the targeting of the nanoparticle to the tumor. So this paper is currently under review, and I cannot share that information. So yeah, thank you. This is a very good question. Yeah.
If you have follow-up to that, and if you want to speak out, just do so. Just unmute yourself.
I think that people are not allowed to unmute, so if you want
okay.
muted, just raise your hand and then we can. That's mine.
The next question is from Siddesh. Would having a very small particle size significantly affect the half-life of nanoparticles in the blood?
Yeah, this is a very the important question. You know, just like the use of antibody, whether you want to use intact antibody or you want to use FAP fragment. You know, like the intact antibody, they will have the non-specific uptake, but the small fragment and has less problem, but they stay in the blood for a shorter period of time. I think it's a matter of the balance of the both. You know, you don't want your particles to be too small because it can be quickly eliminated from blood. You guys probably heard about that most of the nanoparticles, they claim that only have 1% of the dose that can reach the tumor, and some of the studies are done with some very small size particles, so they can have a rapid elimination from the kidney through the excretion. So yeah, I think to answer your question, I think this is need to be experimentally tested in a particular model, and to see what is the underlying problem, you know, for that approach. Yeah. So yeah, I think that's all.
The next question is about, have you studied the endosomal escape of the ultra small nanoparticles and the ones coated with CS?
No, not yet. Yeah, that would be a pleasure for a new student. Yeah, this is a very good question. Yeah, very good. We haven't done that yet.
So I have a question, if I may. Is that all right,
Sure,
Sean?
absolutely. Yes.
Awesome. So first, I just want to congratulate you on this outstanding work. It's brings back so many thoughts of my graduate school education and all of that, looking at trying to optimize tumor targeting and showing spheroids and tumor models. That's outstanding. It kind of looks like your work is challenging the EPR effect, right? That you have this like active transport that's aiding in the accumulation of these nanoparticles at the tumor site through the transcytosis mechanism, right?
Yes,
That's
yes.
probably like this intimate balance between optimizing the interaction with those
Yes.
tumor endothelial cells while
Yes.
still leveraging some EPR effect. Can you kind of let me know what your thoughts are on that?
Yeah, I think the EPI is still a hot topic of debate. You know, this is also a challenging for us to face. You know, when we submit a grant proposal, they said EPI doesn't work, and your grant cannot fly. So this is a question. But on the other hand, this is also a real question. You know, and we need to address. And so whether EPI exists in the human patient requires more study, but at the same time, we do need to develop a strategy to deal with this problem if, you know, we don't have significant EPI in the human patient. Yeah, particularly for the size that is relatively large, about 100 nanometers, that cannot benefit from EPI, you need to think about other approach. But for the ultra small size of particles, we study in my work through a different mechanism because the size is very small and they can reach they might benefit from EPR. But after they reach the tumor, you also have the retention and also the penetration and the issue. So that's a question that I asked by one of the audience, but we actually found out our ultra-small sum of particles does not simply benefit from EPR. It actually also have an active targeting and component. Yeah.
It's a really neat system that kind of shows results contrary to the conventional like size limitations.
Yeah. I still remember I think it's okay and to share this because this is not confidential. I once served a study session with Dr. Li Huang, and one reviewer said the EPR doesn't work, and Li said, well, if you see that comments, I think we need to triage the 50% of the grants, and we can go home early. Yeah, exactly. This is exactly the comments he made. I think it is a reasonable comments because our field is still evolving. There's a lot of questions that we need to study, and you need to deal with the specific question in the proposal. You cannot kill a grant based on this. These very general comments, which is not good. Yeah.
Makes sense.
Yeah. Any other questions? There's one more question from Mohammed. What are the immunogenicity or toxicity profiles of these nanoparticles?
Our limited studies have shown that it's relatively safe, does not cause the significant and immune response. And those particles that are removed probably will be the sinusoid endothelial cells and also some Kupffer cells and for some reason these cells are quite immunosuppressive. Yeah, so we did not see a lot of toxicity. And also, we also realized the backbone of the polymer that are published in our paper is not biodegradable. So that's not going to move cannot go too far. So currently, we are trying to develop a biodegradable backbone, but still have the general design of the nanoparticles. This is what we are working on right now. Yeah.
So the next question from my side, so it's more general question for the field, I guess. What does the future hold for these wonderful discoveries as chemotherapy failure still remains with us?
Yeah, we don't know. I think only clinical studies can tell. There are so many wonderful stories, from animal studies, but not so many successfully translated into clinic. I talked to a lot of the clinician, and they told me that the tumor biology in the human patient is very, very important. A lot of times, whether something can be translated into clinic is dictated by biology in the patient. We definitely want to see nanoparticles important, but also feel like the study of the biology is also equally important here.
Thank you. Last question, Sri Harsha. Does anti-PEG antibodies have any inhibitory effects for regulated mice cells?
Yeah, this is also a big question in our field, and we didn't look at this, so we don't know. Yeah, we don't know. But it is a very important question. Yeah.
Okay. Yeah. I think that's the last question. Anyone else?
Yeah, thank you. Yeah, this is all wonderful questions. Yeah.
Thank you so much, Dr. Lee. This has been really wonderful.
Thank you.
Thank you so much for doing that.
Okay. So nice meeting all of you. Yeah.
Thanks everyone for joining. We'll see you next time. And thank you again, Dr. Lee.
Okay.
Bye.
Recording.